talked a little bit about muscle tissue, and we talked about our different tissues. The muscles were one of them. Today we're going to talk more in depth about them. And we have about 600 skeletal muscles. 2 weeks from now in lab will be the muscles lab, and it's going to be another doozy. There's going to be a whole bunch of muscles to memorize. Luckily, we don't have to memorize all 600 of them. But muscle makes up about half of our body weight. And we learned that there were 3 types. There was skeletal muscle, cardiac muscle, and smooth muscle. What would you say is different with skeletal muscle compared to the other 2? It's voluntary, right? Skeletal muscle is voluntary, cardiac and smooth are involuntary. If you look at these under the microscope, how are you going to tell skeletal from the other 2? Striations. So skeletal muscle has very, very distinct striations. They're really large cells with multiple nuclei. Muscle tissue as a whole has one purpose, and that is take ATP as a source of energy and convert it into motion. So it is going to take a protein and it's going to burn energy and move that protein over. And that protein is going to be attached to something. So when you move that protein, it's going to move the muscle as a whole. Study of muscles is myology. So muscles have a whole bunch of functions, all related to that movement principle. So there is movement in terms of moving my body. There is stability. Muscles allow me to stand here without falling over. It allows me to control the openings to my body, like my mouth, my eyes, things like that. Muscles produce a lot of the body heat that we need in order to stay alive. And then the muscles are also going to help control the amount of glucose in our bloodstream. They can suck up some of the excess glucose. Muscles that control the openings are called sphincters. There are a number of them, but the majority of them are part of our digestive system. All right. This is a cross-section of a muscle. And hopefully this looks somewhat familiar because it looks similar to the nerve that we saw in the lab. It's bundled up essentially the same way. We're going to have small fibers bundled up into medium fibers that are then bundled together in a big fiber. We're not going to spend too much time on this figure right now because the next few slides explain the things that are on here. But what I want to point out is that we're going to talk about muscle fibers. The term muscle fiber is a muscle cell. When we talk about muscle fibers, that is one muscle cell. So around each muscle fiber or cell is a membrane called the endomysium. So if we go back and look, this is one muscle cell, that's one muscle fiber, and we have an endomysium going around that one cell. Endomysium is not on this diagram. That is more or less sort of like the plasma membrane of that cell. It has a purpose. It kind of provides room for things that need to be next to that nerve, or sorry, that muscle fiber. And so you can imagine if you pack too many muscle cells in here, there's not going to be any room for blood vessels. And so you need to have this endomysium that kind of goes around each one to allow space for nerves and blood vessels to be able to get through there. And it's also going to control the amount of ions outside of our cell, as well as nutrients that need to get into we're going to see how muscles react. It's very similar to nerves. And you're going to have a big influx of positively charged ions into the cell. So we need to maintain the correct amount of ions outside the cell, and this endomysium is going to help with that. Outside of the endomysium is what's going to be called the paramysium. This is going to be wrapped around what's called a fascicle. A fascicle is a bundle of muscle fibers. This is where larger nerves and blood vessels are going to be. Small ones can be in the endomysium, larger ones need to be in the paramysium. So this here is a fascicle. So this is one cell. We take a number of cells, wrap them up. In the paramysium, we have a fascicle. So that's going to be a bundle of muscle fibers. Outside of that is the epimysium. So we can take a number of fascicles, put those together, bundle those together, and then wrap that in the epimysium. This is essentially going to be around what we would consider an entire muscle. And so what we think of as a muscle is actually made up of a number of fascicles bundled together, and each fascicle is a number of muscle fibers that are bundled together. So going back, this is a muscle, and so on the outside of this is going to be the epimysium. Then there is also the fascia. The fascia is going to be outside of the epimysium. A fascia is going to separate one muscle from another muscle. So it's going to be thicker than the epimysium. So here we see the epimysium, and then out here is the fascia. And so if you dissect something and you see the outside of a muscle, you're actually going to see this white covering on it, and that white covering is going to be the fascia. If we take muscles in a group, the group is going to be multiple muscles that serve more or less the same function. And if we have multiple muscles that work together for one function, that is going to be called a muscle compartment. So what we're looking at here is the bottom part of a leg. So it's essentially a cross-section of a leg. And so we have the two bones here, and then there's going to be three primary muscles. There's going to be two here that you normally picture as being on the back of your calf, and then there's a larger one underneath. Together, that is going to be considered a muscle compartment. Those three muscles work together for the purpose of moving your leg. And so that is a muscle compartment. And then included in that is also going to be the blood vessels in the nerves that go along with those muscles. So the muscle compartment is essentially going to be everything needed in order for those muscles to work. It's possible to have multiple compartments next to each other, and those muscle compartments are going to be separated by what's called the intermuscular septa. And so here we can see that we have three muscles here, but then there are these other compartments up here that are not muscle compartments. They serve different purposes, but labeled up here we have different septa. And so the septa are the big, wide, white membranes here that are going to separate the muscle compartment from the other compartments. Those muscles have to attach to our bone. Muscles exist to move things. The majority of them are going to move bones. So there are a couple of ways that we can do that. We can attach muscles to bone indirectly using tendons. So a tendon connects a muscle to a bone. And tendons are essentially made of collagen, and they are ingrained in the endomysium, paramecium, and epimecium. They essentially just continue out the end of the muscle. So imagine that diagram. We had all of those membranes, and then imagine them continuing up to the end of the muscle. The muscle inside the membrane is going to stop, but the membrane itself is going to continue. It's just going to continue up and become the tendon. And that tendon is then going to attach to the bone. So if the muscle pulls, it'll pull the tendon, and that will make the bone move as well. You don't need to worry about these. So this was indirect attachment. You can also have a direct attachment, where the muscle more or less attaches directly to the bone, and it looks like it's a continuation of the bone. So when we talk about these connections, we have a couple terms that we can use. The origin of the muscle is going to be where the muscle attaches to a bone at the stationary end of a muscle. And this is a little bit subjective, stationary, because if you really think about it, so this is a forearm, right? So we have a forearm down here, and then we have the upper arm here. And so we're talking about these muscles, and we are going to say that one of them is stationary. If you look at this, and you say you're going to flex your muscle, which end are you going to say is most stationary? The shoulder end. Even though, in reality, it's going to move a little bit. It's not completely stationary, but it is more stationary than the other end. And so we say where the muscle attaches to the stationary end is going to be the origin. Moving down, the thick part of in the middle of the muscle is going to be the belly, and then the other end is going to be the insertion. So the belly is easy, that's in the middle. For the origin in the insertion, you have to figure out when this muscle flexes, when it contracts, which end is going to move more. That end will have the insertion, the other end will be the origin. So again, we're going to talk about this diagram piece by piece, so this is just here for me to keep referring back to. But this is a muscle fiber. This is one cell. It gets confusing because this looks very similar to one of those fascicles, right? We have a bunch of bundles inside of it. And so it's essentially the same idea, but this is only one cell. These are nuclei up here. So this is one cell, and these are going to be parts of one cell. So the plasma membrane of a muscle cell is called the sarcolemma. This is the plasma membrane. But because it's a specialized cell, they gave it a specialized name. Inside of the muscle cell is the sarcoplasm. This is the cytoplasm, but they call it the sarcoplasm. The sarcoplasm. In that are going to be a few things. The biggest thing are the myofibrils. These are the proteins, these are the chains that are going to make the cell move. This is what's going to contract. If we go back and look at the picture, that is all of these red bundles. They take up by far the vast majority of the cell. The rest of the cell exists only to allow these to work. So those are going to be almost the entire volume of the cell. Also in the sarcoplasm is glycogen. The majority of the glycogen in our body is stored in our liver. The liver is the bank for this glycogen, but muscles also keep a little bit on hand. You keep most of your money in the bank, but you probably keep a little bit of it in a drawer somewhere, right? You got to have some for quick access. So in the muscle cell is going to be some glycogen, and then there's also going to be myoglobin. Myoglobin is very similar to hemoglobin. What's hemoglobin's purpose? It's in your blood, it carries oxygen. Why then do we need myoglobin if we have hemoglobin? It's very similar, just keeping the glycogen on hand. This is myoglobin that stays inside of the cell. Normally, for a cell to get oxygen, it has to wait for hemoglobin to come by and drop it off, and then for the oxygen to come in. But muscles need a lot of oxygen very quickly when they contract. So they're able to keep some of that oxygen on hand, so that when they need to contract, they have oxygen ready to go. Then they can wait for the hemoglobin to come and replenish the myoglobin. Remember, skeletal muscle, there are multiple nuclei. There are only a few cell types in our body that have multiple nuclei, and the skeletal muscle is one of them. And what you'll see in the diagram and on the slide are the nuclei are pressed up against the inside of the sarcolemma. Why do you think that is?
You have more muscle, you make more muscle cells when you work out and stuff than you would like bones and stuff.
So we've got the nuclei here. Why are the nuclei here and not in the center? Because most other cells, if you look at them under the microscope, the nuclei is going to be fairly close to the center. Why are they not in the center here, do you think? That's okay. You're only on video, it's okay. But you can't see who you are. So you can say whoever you want to be. So why do you think the nuclei are here?
Does it have to do with the energy production and
Simpler.
stuff?
Space.
What about space?
Space saving.
Yeah. There's too much stuff, all the important stuff's in the middle. And so you just kind of stick the nuclei where they fit, right? If you put a nuclei down here, all of a sudden these are going to have to kind of bend around it. And that's not going to work. They need to be straight. And so you just kind of put the nuclei out on the outside where they can fit, and they just, the sarcolemma just kind of bulge and go around. There are also mitochondria, and these are packed between the myofibrils. So here we have mitochondria. Why do you think the mitochondria are not on the outside?
Because they need to provide ATP.
Right. They are needed in order to make the myofibrils work. The nuclei just kind of keep tabs on the whole cell and make sure it doesn't die. The mitochondria, if they aren't working, they're not producing the ATP needed for the myofibrils. And so they need that ATP that gets made needs to very quickly get into the myofibrils. And also, they're smaller, right? A nucleus is pretty big. Mitochondria are small. You can kind of fit them in the little spaces. And so they're not really getting in the way of the myofibrils. The smooth ER of a muscle cell is called the sarcoplasmic reticulum, or the SR. And so this is going to be wrapped around in little tubules around all of the microfibrils. And then when we look at that picture again, we're going to see crossing from one muscle fiber to the other are what's going to be called the terminal cisterna. And the sarcoplasmic reticulum has the primary purpose of holding on to calcium. calcium is going to be very, very important for making the muscle contract. Calcium is going to move into these muscle cells and cause the myofibrils to contract. So you need someplace to store that calcium, keep it kind of locked up so that it doesn't just leak in, but it has to be close by so that when you want it to go in, it's right there. So the calcium is going to stick around in the sarcoplasmic reticulum. So that is going to be this blue network here. That is our sarcoplasmic reticulum. Terminal cisterna are these blue tubes that go sideways. So this is like the main line. If you're plumbing a house, you have a main line, and then you have all the little pipes that come off of it. And so this is the primary part is going to connect all of the different myofibrils apart, and then coming off of that will be that meshwork of smaller tubes that cover the entire myofibril. There's also, right next to that terminal cisterna, a T tubule. So when we go back to the picture, you'll see that there are two of these terminal cisterna and one T tubule. Together, they make up what we call the triad. So we have two terminal cisterna, and then we have one T tubule. The whole word is transverse tubule, and together it is a triad because there are three pipes there. The T tubules actually go between cells. So remember, this is one cell. The T tubule, the purple one, is going to connect all of the cells together, and then it comes in, and then the terminal cisterna are the main pipes within a cell that feed into all the little pipes for each myofibril. Next, we'll talk about the myofibrils themselves. This is where the magic happens. These are what are going to contract and make our muscle contract. And there's a lot of moving parts here. It's like taking open an engine. There's a whole bunch of things that go on in here. The general skeleton of this contains two types of filaments. There are thick filaments, and then there are thin filaments. So big purple ones here are thick filaments. These are going to be our thin filaments. And they're just sort of alternating thick and thin, and they overlap with each other. So the thick filaments are made up of myosin. Myosin is a protein, and each myosin protein looks like this. It's like two golf clubs wrapped around each other. And then the two heads stick up next to each other. And those heads are going to be very important. They're going to grab on to the thin filaments. We then take a whole bunch of the myosin molecules and weave them together to make this thick filament. And because we have the heads on each myosin molecule, the heads are going to stick out from the thick filament. In a thick filament, we have heads on one side that stick to the left. We have heads over here that stick to the right. And then right in the very center of the thick filament, there are no myosin heads. This is called the bare zone. And we're going to see that this has important functions when we actually start contracting our fibrils. Then there are the thin filaments. These are made up of actin, fibrous actin or F-actin, and they're two strands twisted together, just like our thick filaments, but then we don't twist multiple of those on top of each other. So with myosin, there was two strands twisted together, but then you took a whole bunch of those and twisted them together to get a thick filament. Here, we just have two strands of actin twisted together. So the whole fiber is called F-actin. Each little piece is called G-actin or globular actin because it's ball-shaped. So each monomer here is one globule. Put them a whole bunch together and you get a fiber. Sitting on this, we have a couple different molecules. So this big, long, white molecule is called tropomyosin. And hidden behind where these white molecules are, there are little dimples that we're going to see in another picture. Those dimples are what the thick filament is going to grab onto. That is the anchor point of these thin filaments. And so if tropomyosin is covering up those anchor points, the muscle is not going to do anything. And so this is an inactive thin filament because the tropomyosin molecules are covering up the active site. And then in the center of the tropomyosin is a troponin molecule. These bind calcium. What we're going to see is that when calcium gets into a muscle, it's going to stick to the troponin. The troponin is going to pull the tropomyosin off, activating the thin filament by exposing those dimples. We also have enough myofibril elastic filaments. So this is kind of a bigger picture diagram now. So you see we have some thick filaments, and then in between the thick filaments are thin filaments. But the thick filaments are attached to these little springy things here. We have these anchors. The anchors are called Z-discs. They're going to run periodically throughout the muscle fiber. And then attaching the thick filament to the Z-disc is going to be an elastic filament. This is called titin. It is a huge, huge, huge molecule. This is a whole bunch of molecules wrapped together. Same thing with this. This is one gigantic molecule, so they called it titin. This serves a couple purposes. Number one, it's going to control where the thick filament goes. Imagine if you have a filament or a piece of wood or a stick or something, and then you have springs on each end. Depending on the length and the strength of those springs, you can control where that filament ends up. If this spring is longer than that one, this whole filament's going to be moved over a little bit. It's also going to prevent the muscle from overstretching. What we're going to see is that when this happens, everything is going to contract. And when you contract, it's going to pull on the springs over here. So imagine I'm going to take these two Z-discs and move them together. But when I do that, this stuff over here and this stuff over there is going to stay in place. And so these springs here are going to have to stretch to allow this Z-disc to come in. You know, if you take a spring, the further you pull it, the harder it gets to pull. And so as this starts to contract, these are going to start pulling it back and saying, no, you can't go any further. So it's going to get harder and harder to pull. And then when you let go, they're going to pull these Z-discs back to where they started and reset the whole system. If these were not springs, and you contracted this, and then you set down the bucket that you were holding, your muscle would never relax. It would be contracted, and it would stay contracted. Because of the springiness, it's going to stretch back out so that it can lift again. So, now we're going to get into the actual movement. The movement happens between myosin and actin. Myosin is the thick filament, the actin is the thin filament. These are what we call the contractile proteins. myosin and actin. The tropomyosin and the troponin are going to regulate the process. Troponin is going to move tropomyosin, which is going to allow the myosin and actin to do their thing. They are not controlled. If they are able to contract, they will contract. And so you need the tropomyosin and the troponin to stop them from doing that. It's like a kid, if you give them ice cream, they're going to eat the ice cream. You have to step in and take the ice cream away from them. So, like I said, this whole process is going to start by calcium entering the cell. Calcium is going to come out of, what were those tubules on the outside? The blue meshwork. Those are the big ones connected to the meshwork. I'm going to go back a few slides. Where is the calcium stored? What's the name of the little blue sarcoplasmic reticulum? Thanks. And so, the calcium is going to come out of that and come into our muscle fiber. Like I said, that calcium is going to bind to the troponin. It's going to cause the tropomyosin to move. When it does that, the heads of the thick filament are going to be able to grab onto our actin. So this is what the whole system looks like. And so our actin is lined up with our myosin heads. In the middle here, where there were no myosin heads, in our bare zone, there is no actin. And so there's no actin to grab onto, and so we don't need any of the myosin heads there. But we have these alternating thin, thick filaments. Okay, so now when we contract, all this contraction is going to be some protein inside of a cell. We don't care about proteins contracting inside of a cell. We want what the muscle is attached to move. That's the purpose of our muscles. And so we need to connect our filaments down here to the outside world. So down here in our cell, we have our Z discs, and we have our thick and our thin filaments, and connected to the outermost thin filament is a protein called dystrophin. And dystrophin is going to connect this whole system to the sarcolemma, which is the plasma membrane, and then that will be attached to the muscle cells around it so that everything can contract at once. Dystrophin is what's wrong in muscular dystrophy. Muscular dystrophy has non-functional dystrophin. And so if that dystrophin is not functional, what do you think is going to happen? If this is inside the cell, contracting, but the link to the outside world is not there. What? Say it again? Right. The fibers in each cell will just keep going and going and going, but your muscles can't contract. The fact that we have these bare zones, good question?
Before we move on, is that an issue that people have, that they don't have those proteins, or is that something they do accidentally or?
No, it's a genetic. It's not as strong as it should be. And so essentially sometimes it rips.
Is that like sclerosis or hypertrophic lateral sclerosis?
No, that's a very different thing. The muscular dystrophy is a genetic disease where there's just a mutation in this protein that causes a very debilitating disease.
And what if it's shorter?
If it, well, shorter is not necessarily a bad, you can get around that. Because if you move this over here, then you don't have to go as long, right? It's when it's too weak. So it's attached, the muscle fibers contract, and it can just snap.
Because I have a friend whose neck is always being pulled down, and he can't like stretch it out, so it's just constantly pulled down. So I was wondering if that kind of.
No, I think that's probably something else. So if we look at these, there are distinct regions here. And those regions are what's going to cause those striations when you look at the muscle fiber under the microscope. We have Z-disc here. We have here where it is thick filaments and thin filaments. And then we have here in the middle where there is only thick filaments. And so we're going to name these parts of the striations with letters and call them different bands. So the first one is we have A bands and I bands. A bands are going to be dark bands. You look under the microscope and you see the dark stripe, that is going to be an A band. The A band is going to be where the thick and the thin filaments overlap. So where you have the most protein, that's going to be the A band or the dark band. And then there's also going to be I bands or light bands. Where do you think the I bands are in that diagram? Near the Z discs, yeah. There's not a whole lot of protein here. Between the two A bands, we have an H band. It's still going to be pretty dark. These thick filaments are very heavy. It's going to be darker than over here, but it's not as dark as this. This is an H band. So we have dark bands, light bands, and H bands. And right in the very center is going to be what's called an M line. So H band is the center of our A band, and then right in the very center of that is the M line. I band is called an I band because it will actually change the polarization of light. So you've seen polarized sunglasses, right? Polarized light is light with waves are all going in the same pattern. And so because of the proteins here, they actually come together and they're going to reflect polarized light differently than the dark band will. So that's just where that word came from. You don't need to know why it's called an I band. We said the Z disc is sort of an anchor. It's where all of the springs, the elastic fibers, are going to attach to. And it's right in the middle of our I band. So I really hope that you're able to see the different regions here. We're going to talk about the regions on this exam. We're also going to see them again in the lab. I'm sure you'll see them on the lab practical. You need to be able to look at a diagram like this and figure out where those different bands are and tell me what is present in each of those bands. Tell me which proteins are present in, for example, the I band or the H band. So this is an actual microscope picture, not a diagram. And so you can see this light band. You have alternating dark, light, dark, light, dark. In the center of the light band is a very, very narrow dark band. That's going to be our Z disc. Obviously the light band is the I band. Dark is our dark band or A band. Right in the very center is a strip that's not quite as dark. That's our H band. And right in the very center of that is our M line. The region from one z-disk to another z-disk is called a sarcomere. That is one section of this system that's going to contract. And each sarcomere is independent of the others. One can contract while the one next to it won't. That normally doesn't happen because one sarcomere contracting is not going to produce much force. Our brain is going to force many of them to contract all at the same time, but in reality, each sarcomere is our functional unit that we're dealing with. When they contract, the muscle cell is going to shorten because the sarcomeres shorten. And they're all going to shorten at once. And so one band that was this wide, one sarcomere that was this wide, is now going to contract to this big. And if you take a whole bunch of them and do that, the muscle cell that was this big is now this big. A muscle, which is multiple cells end to end, is going to go from being this big to this big. So the muscle is going to shorten. So whatever is attached to the ends of that muscle, they're going to have to come closer together, right? That's what's happening if I'm bending my arm. Muscles are contracting, pulling things closer together because the muscle is getting shorter, so the bones have to go with them. It's important to remember that none of these filaments, the thick or the thin filaments, actually change length. They just overlap a bit more. I mean, you can overlap filaments like that, and then you can overlap them like that. My fingers did not change length, but the distance between the palms in my hand did. And so just by taking the thick and thin filaments and sliding them over each other, contracting them, the Z-disc on each side will come closer, your sarcomere will shrink. When that shortening happens, dystrophin, which is attached to the outside, is going to get moved. If what's attached to the dystrophin on the inside moves together, what dystrophin is attached to on the outside is going to have to move as well. So the force gets transferred to whatever is outside of that cell. I'm going to skip these. These are just what we talked about. This is kind of an overview in table format. I think this is a good time for a break. Let us take 5 to 10 minute break. I have your exams from a couple weeks ago. If you want to look at them, I can show them to you. Remember, I can't let you write anything down and take them back to your seat or whatever. So you're able to look at them and get them back to me. we're going to work our way through the process of this contraction actually happening. There's going to be 4 main stages that are going to happen here. And so every time you contract, you also have to relax. It's a cycle. You can't contract twice in a row because you have to relax in between. The first step is going to be excitation. This is going to be caused by nerves. This is going to be induced by changes in membrane potential. So electrical changes are going to happen, and this is what's going to be called excitation. There's excitation, contraction, coupling. So that's the third step is contraction. So the step between excitation and contraction is excitation, contraction, coupling. And so this is going to be the quick little burst of events that are going to happen between the nerve firing and the contraction happening. This is not going to take very long at all, but there is an extra little step in here called excitation, contraction, coupling. We have contraction, where the Z discs come closer together, the thin and thick filaments are going to slide over each other and contract. And then the fourth is going to be relaxation. And so whatever stimulated the initial excitation will be gone. The contraction will have happened, and then the myosin will let go, and everything will come back to where it started, and then we can go through the entire process again. So we're going to start with excitation. So what you're looking at here is there is a sarcoplasm here. So this is a muscle cell down here, and then we have a nerve attached to it up here. So we have a synaptic knob. That's the end of an axon. So this is an axon coming off of a neuron. And so the signal is going to come down our nerve, and then we're going to have these vesicles, like we've seen before. And the vesicles are going to contain acetylcholine. We're going to abbreviate that ACH. Where's the... There's the acetylcholine is right there. That's a small molecule. It's a signal molecule. So it's going to bind to a receptor down here on the muscle cell. So we have ACH receptors on the muscle cell. And so the space between the axon and the muscle cell is a synapse, just like when we were connecting one neuron to another. Synaptic vesicles are going to dump the ACH into the synapse, and some of it is going to bind to the ACH receptors on the sarcoplasm. Not sarcoplasm, sarcolemma. When the ACH binds to this receptor, it is going to open, and it is a pore. So when ACH, the little green here, is going to bind, and this pore is going to open. And when that opens, calcium is going to be able to go through it. So we have acetylcholine binding. This is going to open this pore. Not only is calcium going to go through it, but so is sodium and potassium. And so the changing of the sodium and potassium is going to be what is going to cause a change in the membrane potential. That is going to cause a change in the voltage. And this voltage change is what's going to cause the muscle to contract in the end. And so it says we have 2 ACH molecules that are going to bind. It's going to open, and when it opens, sodium is going to rush in. There's a whole bunch of sodium outside that wants to get in, and then there's some potassium on the inside that wants to get out. But it doesn't want to get out as bad as the sodium wants to get in. And so when this opens, the sodium is going to rush in. Sodium is positively charged. And so sodium rushes in, potassium leaves, but more comes in than goes out. And so the voltage is going to change. We now are more positive on the inside than when we started. And what we're going to see is that this is going to cause other pores to open and make this an even bigger change. And so resting potential is where we are here.